All right, let's wrap up the male structures by talking about what's going on the inside. It seems like the simplest thing to do would be to start by talking about where sperm is produced and then how it ultimately gets out of the body. But actually, before I start that, I wanted to point your attention to those spongy tissues of the penis. We can see them in this picture also, right? Here they're colored more of a, lavendery mauve gray color. I don't know. What I want you to notice is how deep they are behind the stomach wall. If you look at where the muscles of the stomach come down, you see this white structure that's the pubic bone, and you see these ligaments that attach the pubic bone to the walls of the pelvic area. So what you're seeing is that the base of the penis is very deeply embedded in the pelvic area. It's not like the penis is just sort of on the outside of the body. It's definitely embedded. About a third of its length is embedded into the body. And that helps with stability during erection. Also, my last piece, I was talking about the engineering marvel that is, human male erections. Part of what helps it to work is that it's really firmly, embedded in the abdomen. I mentioned before that one factor that can affect the appearance of penis length is how much fat is on the abdomen. You can see the fat depicted in this picture with the yellow blobs. The more fat there is, the more surrounded the penis is by the fat. And so, a thinner person looks like they have a larger penis than a fatter person does. And it's just, a function of what else is surrounding it. It's not really anything about the actual penis. So, well, while I'm on this topic, if a person wanted to get, penis enhancement surgery to make their penis longer, the surgery basically pulls this embedded part of the penis out of the abdomen and puts it more out into the world, right? Which can then affect the stability of the erect penis. So it's a pretty risky procedure to try and do that, and probably in the long run not that worth it. So we're looking at a nice cross section. Let's imagine that we're a sperm, and let's start with the testicle. So the testicle is inside of the scrotum, and it is where the sperm is actually created. And there are these tubes inside the testicle that are about the width of a human hair, and that's where the sperm is first formed. And then it has to ripen and develop. And so first there's this splitting of a cell into two parts, and that each part is going to become a sperm, and then it has to mature and develop into a fully formed sperm. So it's a long process, and it occurs as the little sperm is making its way along the little tube that it's in within the testicle. So from birth to moving out of the testicle, it's about 28 days. And then there's a longer waiting period that'll come in a second, but that's the basic process within the testicle itself. These hairs are these little tubules. Like I said, they're about the width of a human hair, and there's, hundreds of them in there, and they're really long. If you took them out and laid them down, they're like hundreds of feet long, each tube. And there's many, many tubes in there, and they're wadded up and stuffed in there. That's like, there's a rhyme to it, but it's pretty much stuffed in there. And so tightly are they stuffed in there that it's kind of under a lot of tension. And again, the testy is covered with sort of a fascia kind of material that keeps the testicle from being able to really swell very easily without a lot of pain. So if a testicle gets struck by anything, it's instantly painful, and it can't really swell very much and without a lot of pain. I mean, it can swell, but it causes that fascia to send out pain messages very quickly and very intensely. So testicles are not really something to be hit, that's for sure. So the testicle is where the sperm is born. It migrates through these tubules, and when it's mostly matured, it'll move into the next structure, which is the thing sitting on top of the testicle that's called the epididymis. Kind of looks like, I don't know, a mohawk hairpiece on top of somebody's head, if you think about it like that, right? You've got your testicle as sort of the face. It's missing a nose or ears or anything. And then you've got the epididymis sitting on top like it's a, I don't know, Elvis haircut, or I don't know whose hairstyle that is. But So the epididymis, again, is full of a bunch of tubes that these sperm have to move through as they're maturing and developing and having proteins added to them, and all sorts of things are happening. And then when they get down to the very bottom of the epididymis, where this tube on the right joins the epididymis, they get into a holding pattern where they're going to wait for the next ejaculation. If no ejaculation comes, then the epididymis will just cause those sperm to be reabsorbed into the bloodstream. So sperm don't have to be ejaculated in order to be removed. As they get too old, they just get reabsorbed into the body, shed through the bloodstream. So sperm will take another probably 20 days to move their way through the epididymis. So it's a pretty long process in the production of sperm, but you'll notice that there's sort of some symmetry. We're going to talk about female reproductive things later. The female sex cells, eggs, there's a 28-day cycle with regard to ripening them. The sperm take about 28 days from birth until they're ready to move into the epididymis. So there's a little symmetry there between the two sexes as far as how they ripen their sex cells. So at the base of the epididymis, there's a junction with that tube that is actually called the vas deferens. So that tube goes from the base of the epididymis up into the abdomen over what is called the ureter that is sticking out from the side of the bladder. So it goes up over the ureter to be hung up there so it's got a nice place to rest, and then ultimately comes down and joins some other structures. So the vas deferens is the tube that carries sperm from the epididymis when it's time for ejaculation. Now, you may have heard of a surgery called a vasectomy, and we're going to talk about that more when we talk about, planning and conceiving children. We'll talk about, birth control and stuff, contraception. So a vasectomy is where you actually cut the vas deferens so that the sperm can no longer be ejaculated. So the testicles still produce sperm, still do all the jobs that they've always been intended to do, but instead of those sperm being ejaculated, they just get reabsorbed in the bloodstream. So that's how conception is prevented. Okay, so we've got the vas deferens going up and around the ureter, and then it joins up with the seminal vesicle. And the seminal vesicle contributes some protein-rich fluid that's going to help to nurture and nourish the sperm on their journey through the female reproductive tract. So the female reproductive tract is quite hostile to sperm, and so the seminal vesicle provides some additional protection so that when the sperm are deposited into that acidic female reproductive tract, they have a chance of survival. So seminal vesicle is going to contribute some fluid. The ejaculatory duct is down a little bit lower where we are inside of this white ball. That white ball is actually the prostate gland. And the prostate gland secretes a bunch of fluid that gets contained, goes along with the sperm and the fluid from the seminal vesicle, and it makes up the seminal fluid. So I always like to ask my students, okay, what percent of seminal fluid, the actual semen, do you think is made up of sperm? And usually I get, guesses ranging from 20% to, 100%. Of course, I've already mentioned that some fluid comes out of the seminal vesicle and the prostate gland. So hopefully you're assuming it can't be 100% because obviously those are contributing something. The real answer is about 1% of the ejaculate is made up of sperm. The rest is made up of seminal vesicle fluid and prostate fluid. Prostate fluid makes up about 90% of the volume of the semen. So the vast majority of what is ejaculated comes out of the prostate gland. About 6 to 8% of the fluid that's in the ejaculate will be coming out of the seminal vesicle. There are some other secretions that can also be contributed that we're not going to bother learning the names of. But so only about 1% is the sperm. So men who oftentimes are concerned about a vasectomy as a birth control method are worried that their ejaculations will be different, will not have the volume that they're used to, that kind of thing. And it turns out that those fears are probably not well founded because so much of the fluid comes from other sources that you usually don't even notice that the volume has changed. So the ejaculatory duct is housed within the prostate gland. Now this ejaculatory duct is super important because do you notice there you've got that Y-shaped area right there in the prostate gland. So all the fluid coming out of the seminal vesicle and then the sperm are entering into the prostate gland where it's going to secrete a bunch of fluid into that tube. And all that activity is convincing the ejaculatory duct to basically flip the switch so that the part of the ejaculatory duct that's coming out of the bladder is blocked. So that even if a man needs to urinate pretty badly, but he's at plateau phase and ejaculation is imminent, he will not urinate. He will ejaculate. If something goes wrong with the ejaculatory duct where it doesn't block the bladder, the error that happens is that the ejaculate, all the fluids that should be expelled out the urethra, will instead be dumped into the bladder, so that the person feels like they ejaculated, but nothing comes out. And obviously that's going to have implications for fertility and other kinds of things. So it's something that can be treated. But when there's a failure, the failure goes the other direction. It doesn't fail in the form of urine coming out of the penis during orgasm. So that ejaculatory duct is super important. All right, so we've got our sperm mixed with our seminal fluid. Bunch of prostate fluid is being combined, and we're at the point of ejaculatory inevitability, where no matter what, ejaculation is going to happen. Even if stimulation changes or something like that, ejaculation is going to happen. So at that moment, the Cowper's gland, see that little ball-shaped thing on the end of a tube? That Cowper's gland is going to secrete some fluid that's going to prepare the urethra for the sperm that's getting ready to come. So it's going to try and neutralize any ammonia that's left in the urethra from recent urination, things like that, and sort of lubricate the urethra. And then ejaculation will occur and the semen will come barreling out. So that's the process, right? It all will come out the urethral opening. So the urethral opening is used for urine and for ejaculation. All right. Well, hopefully that was an interesting trip through the male reproductive tract, pretending that we're sperm and we're on our way out. So I think I hit all the things that could go wrong. I wanted to talk a little bit about that Cowper's fluid. So you may have heard of, or maybe have even tried the withdrawal method as a birth control method. There are a lot of reasons why it's not very effective, and we'll talk about that more when we talk about birth control and contraception. But what I wanted to mention right now is that the Cowper's gland secretes this fluid, and it releases with no sensation or anything. What the man is experiencing when the Cowper's fluid is released is the feeling that orgasm is about to happen. Not that it has, but that it's about to. And so the Cowper's fluid runs down the urethra and gets, clears out anything that might be in there in anticipation of ejaculation. One of the reasons why the withdrawal method can fail is because if there has been a recent ejaculation, and in younger men, this is, fairly common that there might be an ejaculation, maybe a condom was used during that one. And so everything was fine on the first go around. And then now, things started heating up again. And so, but we're just going to sort of spend some time without condom because, it's fun. And I'll withdraw in time, right? And the problem is that the Cowper's fluid can cause any semen or any sperm that might still be in the urethra to be released, be carried along with that Cowper's fluid prior to any sense of orgasm. And so it could be carrying just a couple of sperm, and that's all it takes to cause pregnancy. So one of the reasons why the withdrawal method is not very effective is because, this Cowper's gland can secrete fluid and carry live sperm from a previous ejaculation, that obviously the ejaculation would have happened, today, and probably no urination has happened in between, things like that. But it's possible that that can happen. And so one should never put genitals together that are not protected to prevent pregnancy, because you never know when, a very diligent and determined sperm might make its way out. That's all I'm saying. Okay. I think this is a good time to stop. We'll come back in the next segment and we'll talk about female sexual structures.